my pleasure to introduce Dr. Kelly Bachovchin. I'm sorry for your last name there.
That was good.
Dr. Kelly will be just talking about, giving a discussion and demo of the CDD drug discovery platforms, the BioHarmony and Vault. Dr. Kelly joined the CDD support team to begin her industrial career after completing her PhD in chemistry at Northeastern University. While at Northeastern, Kelly's research focused on synthesizing and optimizing small molecule compounds for the treatment of neglected tropical diseases as part of Professor Michael Pilastri's lab. Before receiving her PhD, Kelly also obtained a master's degree in biotechnology and bachelor's degree in molecular biology, both from Keene University. As a former customer and user of the CDD Vault, Kelly has experienced firsthand how CDD Vault is a powerful and collaborative platform. She wishes to help other scientists to better manage their data and globally connect to further their research as she embarks on her new career at CDD.
Thank you for the introduction. So I'm going to start here on the CDD Vault website page because collaborative drug discovery, being a collaborative platform, is cloud-based. It is a software used to store and analyze and search for data that you are creating in your labs. And so CDD Vault consists of 4 different modules. And although they look like they're separate here, they are all tied together into one platform. So we have an activity and registration side of the database where you're storing your entities and the assay data about those entities that you're storing. We also have an inventory capabilities to track those entities that you are registering. We have a visualization module that you can use to visualize the data that is stored in the vault, along with an electronic laboratory notebook, which is fully integrated with our activity and registration database. So as I was saying, it's a collaborative platform. It's web-based. So in order to get into your private secure CDD Vault, all you would need is a web browser and a working internet connection. So you would log into your CDD Vault with a username and a password, and you can access this vault from anywhere in the world, which is great when you're working from home. So I've generated this search report to show you the different types of data that CDD Vault is used to store. So as you can see, we have full chemical registration capabilities using the Chem Axon cartridge. So you can register your entities or structures with or without the chemical structure. And if you do register with the chemical structure, then there are 20 chemical properties that are calculated based on those structures that are stored. So what the system will do is check these structures at the parent molecule level, so stripped of any salts or solvents, to make sure there are no duplicates in the vault. If the structure is a brand new structure, then you'll get a new molecule created. And if the structure is a duplicate, then you'll have a new batch of that compound created. So this registration, all of this setup of CDD Vault is configurable by the user. And so when a new instance of a vault is set up, the user has the ability to configure and customize their vault to store whatever types of data that they need. So one of the first ways that the system can be configured is through this data container, which we call batch fields. And there's also another type called molecule fields. And so these fields are set up to capture the data about the entities that you're registering. You can see here, we have an external identifier, as well as this PNG image. So these batch fields can store data that are text. You can have numbers. You can also create pick lists to control the vocabulary of what is being uploaded to these fields. You can also attach files, as well as have links to other vaults, as well as other servers. So that being said, the customizable part of CDD Vault allows you to store different types of entities. So I just want to show you a different type of vault that I have here. So this is storing, some plasmids and having the plasmid map. You can capture the sequence information, and obviously there are no chemical structures, but you can capture all of the information about different types of biological entities even. I have another vault here where I have some antibody drug conjugates stored. So you can see here the batch fields are completely different than the one that I had in my small molecule vault. And then the great part about these batch fields is that you can have an ecosystem of vaults and link between each vault. So here I'm storing my antibody drug conjugate and getting the data associated with the conjugate. So here's my dose response curve, but I can also store information about the individual pieces of my antibody drug conjugate in other vaults. Because each vault is customizable, here is the antibody stored in a different vault, and you can see all of the different batch fields that have been created in this particular vault. So you can really set up a whole system to track and, analyze the data around these various different entities. The other type of data container that is configurable by the user within CDD Vault are what we call protocols. And they're really, again, just data containers. Typically, they're used to store biological assay data. And when you're importing your data, what you can import either just a list of numbers, or you can also create calculations based on the data that you import, as well as create these dose response readouts. And these dose response readouts rely on concentrations and raw data that are uploaded. And so they are actually live within the vault. So if I click on my flag outliers and override link, I can actually flag a point as an outlier. Once I save the changes, the curve is refit to exclude the outliers, and then the IC50 is recalculated based on those outliers. And again, just like in the batch fields, these protocols can store data that are text, numbers, pick lists. You can have file attachments as well as links to other servers. So that is an overview of our activity and registration portion of CDD Vault. Or one more part would be the inventory, which I'm not showing here, is tracked at the batch level. So you can actually look up the current and initial amounts of the compounds that you're storing. So when you register a structure, you would register an initial amount, and then you can make debits from that amount to be tracked at the batch level. Uploading data into the database is very simple. Protocols are set up by the users, as I said, so you're not waiting on IT support to come set up a new protocol. You would just create one of the data containers that I showed in the search report table. Then what you would do is choose the file, either an Excel, CSV, SDF file that you want to import into your database. You would select a project, and then you would upload your file. In the second step, you would map the fields in the file to the fields that are created in the vault. So here I can click through and apply each of these fields. I create my experiment run and apply and apply. And now I can process my file. And the last step of my import process is a QC step. So I can check to see what is being imported into the system. As you can see, it doesn't take very long at all. So we have 90 records now that will be imported into this protocol. Once I commit the data, then it's added into the activity and registration database. So while we're waiting on that, I do want to mention that as you saw when I selected that file to be imported, that all of the data within CDD Vault is based on or is associated with at least one project. So projects are how you can silo the data within CDD Vault. You can assign, specific members of the vault to specific projects and really curate who can see what data that is uploaded. And so you can have your CROs be members of the vault as well. And basically, if they are not a part of a project, they don't have the ability to see the data that's in that project. And they don't even know that the data exists in that project because they're not a member. So now that I've imported some data into the vault, I can use this search query to search that data. And this is really the bread and butter. We want to be able to find our data once it's generated and analyze it the best we can. So I can click the green search button now, and it's going to pull up all of the compounds associated with these projects that I have turned on. But I can also search in specific protocols. So now I'm looking at that inhibition data protocol. Maybe I want to look for a threshold percentage for my compounds. But let's say I also want to see the compounds that have been tested in my dose response assay. I can then set a threshold for my IC50 as well. And the more parameters I add into the search query, the more narrow my search results get. So I can also add a substructure to this search query, as well as a chemical property filter. So as I said earlier, the chemical properties are calculated automatically when you register compounds with their structures. So I can put a cutoff there. And then you can see here that this keyword section, you can search by any of those molecule and batch fields that you have created. So if it's something that you want to look for, maybe you want to see a current amount of a compound that's registered in your vault, you can search by any of those fields. So now when I click search, I'll have a more narrow search result. So I have 6 compounds that fit all of the criteria of that search. And you'll see here those thresholds are in bold and the substructure is in green. Anytime I generate one of these search report tables, I have the option to export these data as either Excel files or CSVs, SDFs, and you can even have a zip folder to include the file attachments from the table. I can add my compounds to a collection to go take a look at later. It is a static list of those molecules that can be updated manually, or I can also save my searches. So when I save this search, it will save the parameters of the search query as well as the layout of the search report table. So if I go into my saved searches now, here's my saved searches tab. I can just click search on this saved search, and it will pull up the data that I had searched previously, as well as any new data that has been added to the system that fit the criteria of my search query. So it's a little more flexible and dynamic than, say, adding compounds to a collection. So now I've generated this report table with many columns of numerical data, which allows us to launch these 167 compounds into our visualization tool. It opens up in a new tab. And then by default, you get a scatter plot at the top with a duplication of that search report table down at the bottom and some filters over on the right-hand side. I can customize the axes of this plot. So I can change this to show maybe some inhibition percentage data on the x-axis. I can now look at my mutant 2 inhibition. I can also change the color and the size, so I can map these to different chemical properties, let's say. I can also change this size. Maybe I want to look at my wild type inhibition. And so now you can see I have a 4-parameter scatter plot. that is linked with those filters on the right. So as I click and drag in this space to an area of interest and select the molecules, the filters will also update on the right-hand side. If I select an area in one of my filters, the appropriate points will be crossed out or selected. And then each of these points are structurally aware, so I can hover over a point. If I click on that point, it'll show me all of the information for that particular structure in the search report table that I pulled into Vision. And then if I shift click, I can pin my structure into my plot area, make a nice graphic for a presentation. I can have multiple structures pinned in here. I can change the size. And then what I can do is now save this as a PNG image or as a publication quality PDF. And then the compounds that I have selected in the plot area can be added to a collection back in the vault. And the report table at the bottom can be saved directly as an Excel file from the visualization tool. I can also have multiple plots in the plot area, so up to 4 quadrants, and move them around. And then each of the plots are configurable, so I can change my X and Y axes. And then again, as I select compounds in one plot area, the other plots will update accordingly. If you see the one on the bottom right updating. Okay, so that is our visualization tool. We do also have an application programming interface. So if you are more used to using other visualization softwares like Data Warrior and Spotfire, you can connect CDD Vault directly to those other platforms to visualize your data in a different way, maybe. You can also use the API to actually post and pull data directly from CDD Vault using things like KNIME and Pipeline Pilot to automate your data importing a little bit more. So now the last thing that I'd like to talk about is our electronic laboratory notebook. Again, it is a separate module, but it is just another tab that is a part of the CDD Vault. These ELN entries are also associated to specific projects. So if I uncheck one of these projects, then the associated entries are hidden from view. And I will only see the entries for the projects that I am a member of. You can use the ELN as a traditional notebook, setting up chemical synthesis reactions, taking, your procedure notes on your biological assays, or you can also use it, in really most any other ways, because our ELN entries, when they are created, are just blank pages. So some customers use it as a water cooler where they're discussing their ideas and the compounds that they may want to synthesize down the road as well. And again, because this is a collaborative platform, I can see the entries of the other users in the projects that I'm a member of. So if I wanted to, let's say, copy some reaction conditions that one of my colleagues had used in a synthesis, or duplicate the protocol of a biological assay, I can do that very easily. And it's easily searched. So our ELN has its own search capabilities. So if I type a keyword into the bar here, it'll search the body of the entry as well as any file attachments to that entry. And if those attachments are indexed as text, it will search the body of those attachments as well. So here, this assay is getting picked up in the body of this Word document, as well as in the body of this PDF document that's attached, that's indexed as text. If I refresh my search here, I also have the ability to create some metadata fields for my ELN entries so that I can more easily filter them. So let's say I only wanted to see the entries where I was the author. So that would be my quote unquote notebook. for all of my projects. If I turn off the projects, it would be my notebook for a specific project. And these filters will persist so that if I'm coming into the vault every single day, I don't have to always turn on that filter. I can just take a look at the entries that I'm working on. And then the last type of search capability is the option to search by structure. And I know this comes in handy a lot of times when you're writing papers and you're looking for experimentals, or you're trying to write experimentals for papers, or even your dissertation in my case. You can draw in a structure or a reaction, and it will pull up the entries that have that substructure drawn, or the reaction substructure drawn as well. To get to a specific entry, I would just click on that entry of choice. So here on this particular entry, at the top of the page, you can see you can change the title to be whatever you'd like. This entry is associated with my biotech collaborator project. And then these are the metadata fields that my Vault administrator has created, almost like batch fields, to capture information about each of my ELN entries. So as I scroll through here, you can see, the different types of capabilities within the entry. If I attach a structure that is not registered in my activity and registration platform, I have the option to register it directly into the Vault. If I draw a structure that is already registered in my Vault, then that molecule's name will be displayed below the structure, and then I have the option to register a new batch of that compound. Once I register a batch from the ELN, it actually creates a two-way link back to the ELN entry that it was registered from. So anything that is blue and underlined when I hover over it is a clickable link. So if I click on this molecule name, it will take me to that molecule. And now you can see that batch one was actually registered from this particular ELN entry. And so it comes in handy when you're searching for compounds in the activity and registration platform. You can then see the entries where those compounds were registered from. I can also do this from a stoichiometry table. So down at the bottom here, I inserted a reaction with my little benzene icon, which generates this stoichiometry table, which can be edited. And then I can register compounds directly from the stoichiometry table. So once I'm done with my synthesis, I can put in the mass that I got. It'll calculate a % yield, and then I can register that batch of my molecule. Scrolling back up, you can also attach PDB and mol files, which will be interactive within the body of the entry. That can be done using the insert attachment paperclip icon, or you can just drag and drop files directly into the body of the entry. The great part about CDD Vault being web-based is I could actually take my phone into the lab and take a picture of a gel or a TLC and attach it directly into my ELN entry right there from my phone's browser. Here I have some data files attached. These data files can actually be imported directly into the Vault with this little arrow here. And then once I import the file, it'll change to a magnifying glass, and I can search for that data back in the activity and registration database. And so you can see this is the data that had been imported. If I customize my report, I can turn on various different fields. So I'm going to turn on that run ELN entry. So now I can see which ELN entries that had imported data for these compounds and this particular protocol. We do have an integration with Microsoft Office, so I can expand the size and see a full page document. So I can scroll through the entire document, and then with Microsoft 365, I can make edits in the web that will be auto-saved here in the ELN entry. and the entries autosave 8 times per second, so you don't have to worry about saving your entries. And then finally, we have this assay annotation. This is a mini version of another one of our products coming out called Bioharmony Annotator. And this uses a list of curated ontologies from biological assays that are publicly available. You fill in the various different categories for the assay that you may be running, which will generate the summary at the top based on the categories that you fill in, which can then be copied to your clipboard and used in a protocol or a paper, however you'd like to use that. And then again, these are fully searchable. And to finish up, I'll just say that these entries can be finalized and witnessed. You can export your entries at any time to save a hard copy if you'd like. There's also an audit trail that will track any changes made in any of your entries. And you can also duplicate entire entries so you're not having to rewrite a protocol experiment over and over again. So I have gone very quickly through actually the entire CDD Vault platform. If you'd like a more in-depth demo, you can contact me or my colleague, Pat Deason. We also have a booth that you can come talk to us at if you want more information. And that is all.
Cool. Thank you so much, Kelly, for your presentation. And as she mentioned, you can drop by their booth. You can even book a one-to-one meeting with Kelly, Pat, and her team through our meeting scheduler platform. And if there's any questions on the floor, any thoughts or comments, I'll give everyone a moment.
I'd like to ask take the first question, if that's okay.
Sure.
So I can see where this platform is incredibly useful, like on a day-to-day basis, and it had some really nice sort of graphical interfaces, and as far as being allowing you to present data and things like that. Is it customizable too? If there's some sort of an analytical function you want to do routinely or something, can you add things to it?
Yes, very easily. I didn't have time to show it, but it is very intuitive platform. You're not having to ask me at the support team to set some protocol up for you. You Cool. Really just create a container, import your data, and it's ready to be viewed. It Cool. Takes a couple of minutes.
And then just another thing, this may not be your area of expertise, but one of the things that has been more prevalent these days are there are incidents of biotech companies having their data hacked. Like because when we're talking about just publicly available cloud storage systems, mm-hmm. I imagine you guys have top-notch security in that area. Can you give us a little briefing on that?
We definitely do. So we have our own servers that the data is stored on. There are 2 instances, so we have complete backups of the entire platform. And we have features that we can also turn on for 2-factor authentication, as well as SSO capabilities. So you can use single sign-on to sign into the vault. But we have an entire security team that is monitoring security at all times. And we have extensive security trainings that we go through all the time.
Excellent.
Wonderful. So again, thank you so much, Kelly, for your presentation. We'll be jumping ship now, Dr. Andrea, to our next presenter, Professor Holt.